All right, what is going on, folks? How's it going? You're probably not cooked. You're probably just fine. Hopefully that is the case anyway. My plans for this evening are to answer your questions. I don't have any particular goals as far as going through material unless you just want me to go through material. But in general, I will answer any particular questions that you have, go over any aspects of the test that you want me to go over, like what the test is going to be about or what the format of the test or anything like that. I recognize that not everyone's experience is the same, and so it could be that you don't know a whole lot about the test, which is kind of sad, but that could be the case. So yeah, if you have questions, go ahead and toss them out. Taking a practice test right now. Sweet. Hopefully you've done a few practice tests at this point, or maybe dabbled in some of them, but maybe not. So everybody's got different ways of doing things. So yeah, I'll any questions, any thoughts, or I can just start teaching something if you want me to do that. I'm happy with whatever. Actually, one of the things I want to go over, if any of my own students are in here, is this updated layout for FRQs. It doesn't really change anything, but it doesn't look like any of the FRQs that we've had. And so what does that mean? Yeah, I will explain that in just a moment. That's a good question. So here are some FRQs. This is what the new one's going to look like. And actually, let me go to one that we just recently had in class. Let's see. Wow, there's a lot of practice on this problem, on this exam. Okay, yeah. So here's this one from last year's exam, and I literally just gave this to my students in class, and we did it together. And so notice how it's different. The one I gave you guys, all the business was up here on top, and then all the questions were down at the bottom. And so this one has questions like interspersed. And so notice here, you have this questions after this bit of material. It's because you don't need any content really to answer those questions, and then it gives you some content, and then it asks you some questions about that content, right? And then it's asked you some more questions. And so the way that it's feeding you the questions as you need them, so it's a little more intuitive. So here's another one that we did together as you guys graph this, right? And there's the prompt, and here's the question that you answer from that prompt. And then here's the second part, and it tells you to construct a graph and so forth. And so you get the idea. Okay, independent, dependent, control. So was there a graph on this? Maybe not. If there was a graph, I was going to use that graph to... here we go. Here's a graph. So from this graph, we can actually determine these things. All right. So the CQ value in this particular experiment is what's being measured, right? Because it's on the y-axis. And so that is going to be your dependent variable. Yes, I agree, Ethan. It is so much better because you just had to make sure you get all the questions in is all. That you actually are thorough and you go through the prompt because all the questions aren't gathered in one place anymore. So in this, the dependent variable is the CQ value, which is going to be what is measured. The x-axis in this case is different stages of development, and so that would be the independent variable or what your experiment is based on. You'd have to read the prompt to get the control on this, but the control is just basically your base of comparison. So in your experiment, you're measuring something. What are you measuring it against? And that's going to be your control value. Good. Other questions? Anything? I know some people maybe is afraid to put chat in there so that I understand that as well. I kind of understand that. I know there was a question last night about going over cladograms, and so I can do that. The association of proteins. That's a really good question. I don't know. When I think of it, I know dissociation is like when things come apart, and so I'm assuming. Let's see, association. It's definitely not a term that I'm familiar with. I'm guessing what I think it is. Association. It looks like it has to do with like the way that proteins interact with one another. So that makes sense, right? The way that association of proteins would be like the way that particular proteins interact with one another, or irreversible association of proteins. That actually helps quite a bit. So the irreversible association of proteins is like, so say like, well, let's find a picture. Here's a picture. So here we have an enzyme and a substrate, right? And so this enzyme's probably a protein. And so let's say that this substrate is, I don't know whatever this substrate is. Actually, I have a better picture than this. Here we go. Wait, no. Here we go. Perfect. Inhibitors. And so my enzyme has a particular shape. That enzyme shape determines its function, right? And so the substrate in this particular case is perfectly shaped for the enzyme, and it's this lime wedge shape here. But here we have this competitive inhibitor. Why is it called a competitive inhibitor? Because it's competing directly in the active site of the enzyme. And so this competitive inhibitor may also be a protein. I was actually having this conversation with another student today in 5th period. And so the substrate and the enzyme are designed to come together for a time and then dissociate, right? Whereas the competitive inhibitor is not. The competitive inhibitor doesn't have another little thing that's going to come and break it down later. And so it could cause those two chemicals to be bound together irreversibly. It would be analogous to like getting a tool stuck in a machine and it being unable to get it out because it's like welded in there or something. You know, think about it that way. So that's what I'm assuming it means. I don't know. I don't remember that particular question, but that's what I'm assuming. It probably means something along those lines. Hopefully that helps. All right. Anything else? Any other thoughts or questions, even as you see some of these pictures? Oh, good. I'm glad that helped. I'd have to see the question, but yeah, I'm glad that made sense. I was actually having that discussion today with another student about how, because the question was, well, how does a cell get rid of the enzyme, or how does it get rid of the substrate? And that's how it kind of led to that. What's ETC and photosynthesis and cellular respiration? Good. Let's talk about those. So ETC and cellular respiration, where we go, versus photosynthesis. So let's talk about. Let's find that. All right. So this is photosynthesis. First of all, how do you know? How do I know when I come to this particular thing and I see electron transport chain here, and I know that the electron transport chain is also found in cellular respiration? How do I know I'm looking at photosynthesis? It's these photosystems, right? Cellular respiration is not going to have them. They're not going to have these pigments. They're not going to have any of that. NADPH, remember P? We said P stands for photosynthesis. It really doesn't, but we're going to pretend that it does. Don't want to get pulled over by the prove wrong police this evening. But that P, anytime you see NADPH, you're looking at photosynthesis. And so that's how you just tell the difference when you're looking at a picture. What's the actual difference between them? It's a compare and contrast thing. What's the similarities between them? Similarities is that the idea is that they're both a system of proteins that are going to pass electrons using redox reactions. And because of the passage of those electrons from one protein to the next, it's going to power what we call chemiosmosis, which is the buildup of protons, proton gradient, right? And that proton gradient is going to be used to do some kind of particular work, right? And so that is the, let me find a better picture. Here we go. This is a more complete picture, right? Because it's got ATP synthase in it too. And so that's the similarities. The difference is in photosynthesis, ETC is it's still powering the proton gradient here, but it is not making ATP. Or it's not using oxygen as the final electron acceptor. It is making ATP. There it is right there. And as opposed to in cellular respiration, let's find it real quick. Let's find it. Here it is. All right, so this is the ETC in cellular respiration. Notice what's, it's a good question. I'll get to that in just a second. Notice what's going on here. See these electron carriers? They're dropping the electrons off here, right? And those electrons are powering this proton gradient being made. Here in photosynthesis, the electron carrier is being made, right? NADPH is being made. The electrons are still doing that work, but here the energy from the electrons comes from the production of these electron carriers, which came from the breakdown of glucose. Here, the energy from the electrons is coming directly from the sun because it's being gathered by these photosystems, right? And so in photosynthesis, electron carriers are being made on the ETC, and ATP is also being made using that concentration gradient. In cellular respiration, electron carriers are being used to power the production of ATP. So that's the big difference. They have the same name because they're both doing the same essential thing, carrying electrons, but the way that where those electrons come from, all that kind of stuff is going to be completely different. Hope that was helpful. Do we need to know every detail about photosynthesis, cellular respiration? Just an overview of it. That's a really good question. So let's talk about that, because I think it's actually a good question, because it's, the way that I describe it to my students, it's like, you could really just you could just go to a picture, like, where was that other one? Like this, Krebs here. I only put this picture of Krebs on my slideshow to show the students that it's not important to know every detail, because you could just get lost in malate dehydrogenase if you really wanted to, and you'd have your face staring at it, right? And you'd just be like, okay, well, where does this hydrogen come from, and all that, and you could just get lost. Well, what is that? And you're just cooked at that point. You're like this little boy right here. It's like, just please, just take me away from all of it. And you could memorize all this, but then if you don't know this, this basic picture right here of the whole thing. Let me go back up one more. If you don't know this right here, that photosynthesis, cellular respiration are coupled reactions, right? The products of one or the reactants of the other. If you can't even tell me the equation for photosynthesis and cellular respiration, then you're so that's the idea. It's make sure you have a big picture before you get down in the weeds, because the weeds aren't going to do any good if you don't understand the big picture. And so, when I teach this subject in particular, so I'll give my kids, where's it at? There's a picture. It looks like this, basically. I'll give them this picture and if you just memorize this picture, you'll understand it. So all this, if you can tell me what's going on here, this doesn't really matter as much, right? I would give them these details and talk to them about them, but the big picture is much more important than the weeds. You need to know the equation. You're not going to be asked a question, which of these is the correct equation for photosynthesis? But if you know the equation, you're going to be better equipped to answer questions about it. Memorization is not helpful on this exam, and so like like having a bunch of stuff memorized is not going to ensure that you make a 5. Being able to use that information is the real key. If you're talking about doing practice tests, that's the best way to do that. Like getting lots of practice on those types of questions, seeing a bunch of them. The more questions you see, the better off you're going to be. That rhymed. It wasn't on purpose. All right. What else? Any other questions? I was just actually talking about this picture with a student a couple days ago about how important it is just to kind of like know this basic thing. Like if you don't understand this basic thing, it really doesn't matter. Okay, really good question. So I'll read the question. If you don't have the chat up, how would you approach a long, wordy passage on multiple choice? Should we go through the questions first, read the passage first? How in depth? Okay, so good question. So I'm assuming what you're talking about is like one of those that's like, use this to answer the next 5 questions. I'm assuming that's what Mr. Urali's talking about here. And so what I would do is I would treat it like I treat an ACT passage. Right, and the way that I treat an ACT passage is I don't read it. Right, I would look at the stuff. Like I wouldn't read it because you're going to get a lot of stories. You know what I mean by stories. You're going to get a lot of like wordy nonsense, which is exactly what you're like kind of hitting at. And so, I'll give a caveat to this in a moment. And so then I would just go to the first question and start answering them. The questions on those passages typically play themselves out like an ACT passage too, whereas the first few questions are going to be more of your gimme types, and the questions are going to kind of build in complexity over the course of that particular prompt. And so it may be that you need to go back to the material towards the end of that prompt, and so all of that to say, you may, like me saying that may like scare some of you, and when you get to that prompt, when you get to that page long prompt and answer this for the next 5 questions, look up at the clock, decide how much time you have banked, because you're probably going to have some time banked, and then like, okay, I can dedicate the next 3 minutes to reading this prompt and then answering the question, because then reading that prompt may actually buy you more time, But my first tendency is going to be to just go at each question. And I typically, the way I take these tests is I will read the question sentence and then I'll work backwards, because a lot of times that story is just, it's just too much. But there's risk with that, I don't think you have to take a test that way, but I think you can take a test that way. Some of that's just going to be your ability to process that information. Good question. Really good question. Anything else? Any other questions? I did turn on a sub-only chat because I wanted to kind of make sure that I wasn't getting a bunch of yahoos in here. But it seems like it's going to be pretty tame tonight, too. I was actually a little concerned, but still thinking about watching Mr. Krug's stream and seeing all the just straight craziness that was going on in there. Will you get scrap paper? No, the exam is scrap paper, so you can write in the exam. Now, caveat to that: if you write in the lines on the FRQ, that whatever you write in the lines will be graded. And so if you don't want something to be graded, make sure you strike through it, mark through it with a single line. You don't have to like cover it in a second darkness, but just kind of just kind of strike through it, and they won't grade that portion of it. Good question. Will I go through Unit 5? Yes, I will. I will do that. So let's go through Unit 5. And actually, so Unit 5 is meiosis and heredity, and understanding meiosis is actually the key to understanding all of it, at least for the most part, because this is going to kind of, get the whole idea of homologous pairs and all that business down. And so, yeah, I'll do that. All right, so process that produces, this is some definitions that I've written down. So first of all, thank you there, Mr. Brown, for offering that word of encouragement. Really appreciate that very much. And so make sure you understand the difference between meiosis and mitosis. Mitosis is making identical cells. Meiosis is, the purpose of that is to make gametes, right? To make haploid gametes. And so, and what is haploid? Well, haploid is like having half the number of chromosomes. Diploid's having like both of the pair. So that doesn't really make sense. And so the way I've described it is, it's like shoes. Like you have a pair of shoes. There's a left and a right. They're not identical to one another, right? They're homologous to one another, meaning that they have all the same parts. They're just not identical. And so like you have, like particularly a human genome has 23 homologous pairs. You get 23 from mom, 23 from dad, and that makes up your whole genome, right? The 23 that you get from mom are not identical to the 23 that you get from dad, obviously, because they're not identical people. But they do have all the same genes. So they're all homologous. They may just have different versions of those genes. And so that versions of genes is going to come into play in just a moment, this idea of version of gene, right? And so meiosis is taking that, in a human cell, taking that diploid cell that has 46 chromosomes or 23 pairs, and from it taking and creating and making 4 haploid cells that each have 23, making gametes, essentially, so that they can be donated for sexual reproduction. So that's the process, right? And it's important to note too, I like this picture a lot. Meiosis is not a cycle; it is a process with a product. And so when these gametes here are made, they're like sitting there just chilling, waiting. And that's a zygote on it, I guess, because it's like assuming that there's going to be other ones there. This picture's a little sus because look at that orange in the middle there. It's bad. Somebody did some bad photoshopping. Okay, so this is talking about homologous pairs. Here you go. Oh, I love this picture too because it shows you. So here's this individual is 2n equals 6. And so that means that they have 3 pairs of homologous chromosome or 3 homologous pairs, and they get purple from mom and pink from dad, and they're the same chromosome, same genes, but just different versions. Now, I like that because it shows the different colors. And so this individual is going to make haploids, and in this particular case, they made a pink, light blue, that color green haploid, right? But it could have been different. It could have been purple, that color blue, that color green haploid. And so meiosis is what decides what those haploids are going to be. And so let's talk about that. First part of meiosis is interphase. Interphase is, it's not technically a BS part of meiosis. Just make sure you understand that the DNA is going to replicate. What is this over here? I wonder if I could use that. I'm not going to try. I'm not going to try because it looks kind of sketchy. All right. And then, there's all these phases associated with it. So interphase, big deal here. Thanks for the good luck message. Really appreciate that. Thank you. Really appreciate that. Interphase is going to replicate the DNA. So let me just write some letters here. So I said earlier that we have 46 chromosomes in a particular cell. After interphase, it's going to be 92, right? 92 because all those chromosomes replicated, right? They made a copy of themselves. That was during S phase of interphase. And then in meiosis 1, you're going to have 46 times 2 because in meiosis 1, that big 92 is going to divide in half. The big thing, and so back up here too, This is a diploid cell, right? This is also a diploid cell. It's just got twice the number of chromosomes. Both of these cells are no longer diploid. They're haploid because those homologous pairs have been separated. I'm going to talk about that in just a second. And then right here, you're going to have 23 times 4. And of course, these are all going to be haploid as well. Okay. And so this is just for human genome. If it's like giraffes or like dogs or some sort of like, microorganism, it's going to be different chromosomes. All right. So interphase and then meiosis 1. We didn't really talk about metaphase and telophase because it's not important. You do not get scrap paper. That is correct. And so prophase, big thing to know there is when tetrads form, crossing over occurs, and I bet I've got a picture of that. I don't have a picture of that. Okay, so let's talk about crossing over because it's kind of a big deal. We'll go over here and draw. I'm going to use my Galaxy pen, whatever that is. That's not going to work. I don't like the Galaxy pen. Do I not have other colors? Oh. Okay, here we go. I'm just going to use red. And so here's red, and then the homologous pair for this is blue, not Galaxy. And so this is a homologous pair, right? During prophase 1, this homologous pair is going to literally like line up like this. Like all the homologous parts are going to line up right next to each other. Like when I put my fingers together like this, see all the homologous parts. My hands are not identical to one another, right? They're not identical at all, but they definitely have homologous parts to them, and they're all lined up perfectly right here. And so this is called a tetrad. Why is it a tetrad? Because there's 4 pieces, right? Let me get rid of Galaxy, right? There's Galaxy. Sorry, Galaxy. There's 4 pieces. What are these individual pieces called? Does anybody remember? These are called sister chromatids, right? And they're identical to one another. Red and blue are not identical to one another. And so you have 4. Prefix for 4 is tetra, so these are tetrads. And during prophase 1, crossing over can occur. What is that? Well, it's where these homologous parts separate. I wonder. There it is. Okay, good. It's where these little parts, because they're homologous, right? The same genes are on them. This red is now going to have a piece of blue on it. And this blue is now going to have a piece of red on it, right? And so what is that going to do? It's going to increase diversity in that organism, right? It's going to increase genetic diversity. It's going to give a lot more kind of combinations for things, which is good. So we talk about sexual reproduction allowing for more genetic diversity. This is one of the reasons why that is. Okay, and so that's prophase 1. That's the only time that's going to happen. Another important part of this, I just opened Discord. Whoops. Okay, another important part of this is anaphase 1. What happens during anaphase 1? Homologous pairs separate. And so what is this? And so well, let's go back over here. All right. So in anaphase 1, homologous pairs are going to separate, and what that means is these 2 things that I have here, this thing and its new piece, is going to separate from this one. I wonder if I can group this. I sure can. Maybe not. Okay, it doesn't matter. And so they're going to separate. And so this is going to go to one cell, and this is going to go to another cell. We're going to make these a little smaller so they're more manageable. All right. And so now you can see how these 2 cells that have these things in them, these chromosomes in them, are haploid. Why are they haploid? Well, they've only got half the number of chromosomes. They only have the left shoe, or they only have the right shoe, right? They don't have both. They don't have both mom and dad's complement. They have just mom or dad's complement, right? And so that's anaphase 1. Anaphase 1, homologous pairs separate. Common mistake here, common mistake that students make is to do this. where they think that this is what's happening. Right? And if you do that, homologous pairs have not separated. If your mom was a carrier or not. Well, I'll get to that in just a moment. That's a good question. I'll get to that question. Good question. All right. So in anaphase 2, what's going to happen? In anaphase 2, sister chromatids separate. So what is that going to mean? That means that these individual pieces right here are going to separate, whereas this one's going to go here, this one's going to go here, this one's going to go here, and this one's going to go over there. And then you have 4 haploid gametes. So that is meiosis. And that is going to be pretty much telling you about how gametes separate, or how alleles separate too when we get to that. Because if red is say, big A, and blue is say little a, among other things, you can see how this individual who has a big A, little a genotype has the equal chance to pass big A or little a, right? And so that's kind of tying it all together there. Question about genetics on a problem. They didn't specify if the mom was a carrier or not. What do I do? I'd probably need a little more information, but if it doesn't specify if they're a carrier, then they probably don't have the dominant trait, or it's wanting you to look at the phenotypes of the children and then go back and backward solve the genetic makeup of the mother. That is my guess. We talked about differences, meiosis, mitosis already. Okay, good. So any questions on meiosis? I think that was good. I think that was helpful, hopefully. Good. And a couple more things here. There's crossing over. That was about sex-linked traits. So yeah, you would be able to tell if they were a carrier or not by, well, first of all, if this person has a son that has the recessive, the dad has the disease, but the mother, is it a dominant a disease or a recessive disease? Does it say? Because that would definitely make a difference whether or not. Because you can have dominant sexually disorders or recessive. What do you do if you run out of space in the FRQ? Then you take note of that. Tell yourself that you've written way too much and try not to write as much on the next problem. Because they give you way more space than you'll ever need. You're just literally not going to run out of space. If you do, you've written way too much. This is not an English test. Just answer the question and move on. I'll go over that in a little bit because it's really easy to feel like you have to explain yourself, but they'll only ever ask you when to do that. Otherwise, don't explain yourself. Just give the answer. It's recessive. So what about the children? Does it say any of the children have this disease? Then you'll know. Actually, if either one of the children have the disease, then the mom will be a carrier. That gives. So yeah, that's a really good question, Ethan. What's the easiest way to determine? Is by looking at the what the inheritance pattern is. See if there's any patterns for males in particular, like if neither parent have it and only the males have it from then on, is a good way of thinking about it. Or if the dad has the disorder and the daughters, the mom doesn't have it and the daughters don't have it and the sons have it too, then it's probably autosomal, because dads don't pass X chromosomes to their sons, and so that's it's kind of hard. You need a little more information. But I would look for something. The way that I would do it is I would look at the children and then find an instance of something that could not be true if it was sex-linked. This cannot be true if it's sex-linked. And if you'll find that it's there every time. Find the thing that can't be true if it's sex-linked, and when you find that, you'll know it's autosomal. If you don't find that, then it's probably sex-linked. That is the way I would go about it. And if that doesn't happen, then there's not enough information to answer the question, which isn't going to happen either. It's going to be plain. It'll definitely be plain. Good question. And you can almost guarantee you're going to see a pedigree on this thing. Like, you just almost guarantee it. You're going to see a pedigree. Tuning in from Cookout. Sweet. Cook and out. I feel like Amalika needs to take AP Bio. Just throwing that out there. How much success will be computational? I think it's a pretty small percentage that is going to be like actual problems. Like, think about the practice problems, which I think are the practice exams that you guys took, which I think are pretty good indicators. And it was literally, how many problems did you have to get your calculator out on those 60 multiple choice? It's like maybe 4. So I'm guessing it's going to be like that, like 1 in 15. What are the words related to crossing over in synapsis? Well, synapsis is one of them. Tetrads is what I say, but I've seen this word synapsis more, which synapsis is just the combination of homologous pairs. It's another way of saying it, because that SYN is together, right? Recombinant chromatids, they're recombined. That's what these chromatids that have crossed over are called recombinant chromatids. The ones that didn't cross over are called non-recombinant chromatids. What else? Oh yeah, like map units. Let's see if I've got that on here. I bet I don't. I don't even know where it would be. It's just not something that's covered a lot. Yeah, okay, we did talk about linked genes. Yeah, linked genes is something too. You know what? It's really funny. I was describing this problem today to somebody, and I think I used these exact 2 numbers. Yeah, you do. Which you do get a sheet of what am I thinking? Formula sheet, correct. Which units do you suggest I review the most? Unit 7, according to AP Bio, is likely to have the most questions associated with it, which is evolution. And second to that would be a tie between Unit 3 and Unit 6, which Unit 3 is photosynthesis, cellular respiration. Unit 6 is genes, or like DNA and that sort of thing. So, and it's funny because Unit 6 is my most popular review video right now. People are like wearing it out, but no one's really watching my Unit 3 videos. Maybe they need to go and watch them. So, yeah, linked genes, map units, which is a map unit is like the distance between the percentage of crossing over. So what does that mean? G and A cross over 40% of the time, and so that's map distance is 40. I just literally haven't seen it on exams. Maybe I'm, I don't think I've seen it in anybody. No one's come to me with a map unit question at all on their practice exams. Some of you guys who have taken and answered 150 questions by now, you can tell me if you've seen a map unit problem, but I don't think they exist. It's in the CD, but I don't think they exist. I think they're just shying away from that. Linked genes is definitely in there. And what is linked genes? A linked gene is just linked genes that are located on the same chromosome, and they're more likely to be inherited than not. And so here's this rule down here, which is a good rule. that I'll circle in giant blue pen. Linked genes have a recombination frequency of less than 50%, meaning that they're less likely to recombine. If genes aren't inherited together more than 50% of the time, they probably aren't linked. So it's a good rule of thumb that you might need for 2 questions on the test. But still a good rule of thumb anyway. There's map distance again. There's map distance. For something I don't think is on the test, I dedicated a whole lot of slides to it. Perfect example. Here's a sex-linked pedigree. So let's look at it. Actually, this one's kind of bad because it shows you the ones that are carriers. But anyway, but like Victoria is a carrier, right? And she didn't. You can go and look at all of her sons. She can't. She could pass it to her daughters as well, but in order for her daughters to have it, Albert would also have to have it, right? And so that's not the case here. And then you can go here too. Notice here, Leopold has it. Whatever her name is doesn't. And notice none of the sons do. This isn't a son. These 2 aren't. Because where did the boys get their X chromosome from? From their mom. That's why this little girl is a carrier, because Dad's going to give her one of those X's, and he only has one to give, and his is going to be the bad thing. And so that's the kind of reasoning that you walk through on that problem. Answering multiple. Yeah, I can. Like I don't have any specific questions because we're not allowed to like put College Board stuff up on the board, but and there's not really any release stuff other than really old exams, and it's just not worth using them. But I do have some FRQs, but I don't have any of their release stuff because we're not allowed to show it. So, but if you have a particular question, you can put it in the chat and I can look at it. But otherwise, I'm not able to share that information. We're not, even though we're not supposed to. I know that teachers all over do. Transcription factors. I can do that. Let's go find them. Man, look at that handwriting. Transcription factors. I wonder if it's on this one. No, it's not. So transcription factors just they're proteins involved in transcription. And I know that sounds really ambiguous because it is. We didn't get really any of the names of those proteins other than one. And so, yeah, I'll do case selected populations. Yep. And so the only transcription factor that we were given was RNA polymerase, but there's this whole like transcription complex that is associated with transcription. And you can imagine all these little other proteins that are like tagging along with RNA polymerase, kind of like those little fish that swim beside sharks. But there's nothing specific about them. Nothing specific about them. Case selected species. I don't know what practice exams is. I bet it's here. There's some bears. There's some more bears. Ooh, I love that picture. All right, so first of all, let's talk about exponential growth. So in order to talk about case selected species, we have to talk about exponential growth. And so exponential growth is when a population basically is growing without any restraints, right? It assumes ideal conditions. There aren't any factors that are limiting that population, is another way of thinking about it, right? And so as soon as a population is limited, that's a good question, Alex. I will answer that question for sure, because that kind of was related to the question that you asked earlier. So I will definitely, I think that's a really good question. Actually, one of the more important ones, because my own students tend to write way too much. So exponential growth, no limitations. What if there is a limitation? This population is no longer going to grow like this, right? And so it's going to start to be limited. It's going to show that. Oh, wait, here it is. It's the wrong one. Blah, 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 blah. Look at that picture right there. I love this. I love this. This is where I should have been. And so a K-selected population is a population that is affected by some sort of factor, either density independent or density dependent factor. And that factor is going to basically, it's going to adhere to some sort of carrying capacity, right? And so the carrying capacity is literally K, the variable K, which is why it's called a K-selected species or a K-selected population, because it's going to tend to adhere to a carrying capacity. It's going to be limited by something in the environment. Typically, a prey species is going to be what we would call a K-selected species that is easily limited by something that is in the environment. However, if you take those predators away, then it can grow exponentially. White-tailed deer, perfect example in this part of the world, because we've killed all their predators, and so they just grow exponentially. And so, which is this whole other thing. And this is the equation that you have with logistic growth. And so you'll be given a carrying capacity K, which is usually going to be given. I'm not seeing a problem that wants you to solve for K. It's just going to be too messy, right? And so it's usually going to ask you to solve for rate, which is over here, or it's going to ask you to solve for R max, which is the maximum growth rate. You're not going to be asked to do any kind of crazy algebra or anything. It's just, can you solve for X like an 8th grader? This is basically the extent of what you're going to have to do on these kinds of problems. Here's like a practice problem. I think this came from the one of the AP things, AP videos. All right, so let's look at. It's a really good question about that. So I'm going to pull this one up because I think this is helpful. Okay. So this is a question that was on the exam last year. Yeah, this is last year's exam. And this is the graphing question. So this is the second question. Graphing question is always going to be the second question. And this question says, describe the role of the inner mitochondrial membrane in cellular respiration. You literally do not need to read this in order to get this right. It's just a straight. Some of you guys are really good at content memorization, and you're going to kill on questions like this because it's literally asking you to describe what is the role of this. It's just like rote. And so what is the role of the inner mitochondrial membrane? The role of the inner mitochondrial membrane is to it's the location of the electron transport chain, and the purpose of that is to build a concentration gradient for the production of ATP. That's what I would say. And so that was a one sentence, right? I didn't go into an entire diatribe about what cellular respiration is, right? I didn't tell them how I feel about things. I just answered this simple question, right? What is this? What is the role of this? And I did that, right? That was it. Just to build a concentration gradient for the production of ATP. And that was it. So let's look at this. This is the constructor graph, so we're not going to do this. But however, look at this thing. So this would be a bar graph, right? Because it's categories, or it's comparing one species to the next. Determine which species shows a difference in the number of mitochondria between normal and elevated levels of CO2. All right, so this question is, you can't answer this question until you have the graph, and it's going to use the error bars, right? And so you're going to have so I'll go back to my Unit Z video here. You're going to have some error bars. Let's look at, okay, this is perfect. So the question might be determine if there's a difference between 4 and 5. Or no, let's not use 4 and 5. Let's use 12 and 13, because 12 and 13, you're like, oh wow, there's a big difference between 12 and 13. And so you'd see this question and you'd be like, determine which species show a difference. And you'd be like, well, 12 and 13 show a big difference because look at them. But they don't, because their error bars overlap, right? And so that one's not. But 11 and 12 does show a big difference. So does 11 and 13. There's a big difference between 11. Not a big difference, a small difference, but there is a difference between 11 and 13. There's no difference between 9 and 10 because their error bars overlap. And so that's how you answer that question. And so how do you literally answer this question? Determine which species show a difference. The two species that show a difference are 5 and 6, period. Move on. Don't tell them why you think that. They're not asking why you think that. If they were asking you to tell you that, they would use this word, justify, right? They're not justify why you think that. Didn't say that. It just says determine. The difference, the two species that show a difference are 4 and 5, period, moving on. Don't answer the question any more than that. The readers aren't going to read more than that, right? They're not. They're going to find the right answer and they're going to move on. So you write the right answer and move on. Based on the table, describe the relationship between levels of CO2 and the average number of mitochondria. Can't do that without the graph. So let's see. Predict the phenotype of the leaves of offspring produced from this cross. Let's see. Let's find a different one that doesn't require us to really. Okay, this is perfect. So this is the sand lances one that I talked about last night. Describe the effect of increased biodiversity on the resilience of an ecosystem in a changing environment. I don't even need to read the prompt for this. Describe the effect on increased biodiversity. The effect of increased biodiversity will increase the resilience of an ecosystem in a changing environment, period. That's the effect. It's not asking for you to say why you think that, right? 42 out of 60 is good on multiple choice. That's actually really good. That's probably like a 4, is my guess. Maybe getting close even to a 5. I don't know the exact percentages because I don't keep up with that business. But, yeah, I think that's about right. That's pretty good. Let's see. So, yeah, perfect example, sand lances. Let's see what else. Describe the role of chlorophyll in photosystems and plant cells. Absorb light. The role of chlorophyll is to absorb light. That's it. Don't say more than that. Well, I want to, no, just don't. Just be done. Just walk away. So that's some examples. Good question. Any other questions? I don't even know how many people are watching. Or if anybody's watching. There's a lot of people watching right now, actually. So yeah, if you have questions, if you haven't asked anything, please feel free. No, there aren't bad questions. The only bad one's when you don't ask. Let's see. Actually, there were some questions that came up in this Facebook group that I'm a part of that was asking about like incomplete dominance. Let's see if I've got. How the Barron's books practice exam. So my guess is that those books are going to be, they're definitely more helpful than not doing them. But I don't know. Like I've never looked at one. Like I looked at one years ago. But you know, for me, what I've done all year is just give my students like College Board questions, so they've been exposed to like literally hundreds and hundreds of questions by this point. And so, and I think Khan actually, you know, if you're for like looking for something to use like last minute, I think Khan actually has some pretty good questions. My students have had some luck using Khan as well. But I mean, my guess is that the Barron's books, like they're a business and they want to have a good business, and so they probably have aligned their questions pretty well with the exam. So I would imagine that they're going to be helpful. Definitely better than not doing it, for sure. So, good question. Maybe I need to get one and like look at it. I just don't typically do that. Good. Anything else? Any other like content type questions? How much time should I spend on a question before moving on? Man, it's good. Especially on, well, okay, so let's talk about multiple choice and then let's talk about FRQ. So there's 60 questions, 90 minutes. So that's 90 seconds per question, right? Minute and a half. If you find yourself like having to read the question multiple times, just move along, right? Yes, I can definitely talk about cellular energetics. Just move along. I would skip it, or don't answer it. Come back to it after you feel like you have more time. It could be that another question on the exam will actually kind of like jog your memory a little bit and help you think about it. That happens sometimes. Or if you read it and you know that whatever you think you knew about that content is gone forever, then just literally pick a letter and use that letter the whole time. But yeah, you can't like agonize over a single question. It's just not worth it. Was it? Man, I don't know. That may be somebody for like someone in the chat, because I've heard people talking about it today, like percentage wise, and there's probably calculators online. Yeah, it makes sense what you're asking totally. Like there's percentages. Like I think the percentage of correct answers to get a 3 is like 46%, or that's what it was. And so the way I'd instruct my students is like play to your strengths, right? Like I have some students, for instance, that are like going to kill the multiple choice because they're just really good analytical thinkers, and they're going to kill the multiple choice, and they're actually going to struggle in the FRQ. And so I tell them, pass the exam in the multiple choice and then try to get your 4 or your 5 in the FRQ. And then I have students who are the opposite, struggle with the multiple choice but are really good with content. And so it's the same thing, just play to your strengths on that. But I would use a scoring calculator to kind of figure that out and toy with it. Yeah, I have that same problem. I get tired after like 10 questions, and I just want to quit. So, but I'm old too, so 70% to get a 5. Yeah, that sounds about right. And they adjust that based on, but it's not going to be adjusted a whole lot. Cellular energetics. Talked about this a little bit earlier. Like, do you have a specific question about cellular energetics? Or do you just want me to give like a broad overview? Because I can give a broad overview for sure. Graphing to use a specific measurement. So good question. So when you're graphing, you will want to scale your graph according to the numbers. And so that's a really good question. Let me go back here and like show you this one real quick, because I think it's helpful. Because this is actually, this question is actually pretty good to help you kind of understand scale. And so what I tell my students is, so first of all, what is Y, what is X? Species is the X-axis you're going to put along the bottom. This is the Y-axis, whatever these things are, mitochondria, normal CO2, and elevated CO2. And then you're going to find the low number. So first of all, count your boxes. Yes, I can talk about those things. Alex will do. So you count your number of boxes. And so like, let's say you have 25 boxes, right? And then I'm going to find my lowest number. So it's 0.06 and then plus or minus 0.06. So pretty small, right? So pretty small. Actually, no, it's 0.03 and plus or minus 0.03. So pretty small. And I'm actually going to probably just start this one at 0. And so this is actually a myth. You do not, the bottom left-hand corner of your graph does not have to be 0, 0. And you do not have to draw the little lines to show that you're skipping a bunch of numbers. Whoever teaches that is just like, seriously, just stop. It doesn't have to be 0. It can be whatever you want it to be, right? Now, you don't want your lowest number to be 0.3 because, well, this is plus or minus 0.03, and so you need a little room. So you could start at 0.2 if you wanted to be your bottom number. And then what's going to be your top number? 1.3, 2.4. It's actually going to be 2.7, right? Because 2.4 plus 0.22 is 2.62, and you want a little more wiggle room. And so you're going to do everything between 0.2 and 2.7, which happens to be 2.5. If I've got 25 boxes, each one of my boxes is going to be 0.1, right? Sounds like a magic. And so that's kind of the way that I think about graphing. You're just thinking about that scale, and that way when the grader looks at your graph, you've used the whole thing. It's perfectly scaled out. Everything looks nice and pretty. You don't have your whole graph in the bottom corner or anything like that. So that's the way that I think about it. Good. Fermentation. Let's talk about fermentation. Where is it? Error bars have to be exactly accurate. Yeah, so on that last problem, it's going to be really hard to look at to 0.03. Good question. Do they have to be exactly accurate? No, that's not possible when you're drawing with a pen in an exam. But I would make sure that they are at least the same, meaning that the top, the plus is the same length as the minus, because if they're not, that's just automatically wrong. And that they look appropriately scaled, right? So if I go back to this graph and I have 0.4 and it's plus or minus 0.05, then it's not half of the block. It's just a little bitty portion, right? Just a tiny portion. But it's going to be really hard on your graph to distinguish between 0.05 and 0.07. It's going to be hard for you, and it's going to be hard for your grader. And so just make sure that it makes sense, right? And make sure they're the same. It's going to be really, this is a weird graph, just straight up. The error bars on this one are weird. It's just some of the choices they make, I just don't like, but whatever. They didn't ask me what I liked. All right, so let's talk about this fermentation. We'll start with fermentation, then we'll go through the steps of cellular respiration. So first of all, cellular respiration, the purpose of it is to break down glucose to make ATP. That's the purpose of it. Has to be in the presence of oxygen. If it says to do, yeah, so you could do that. Standard error by 2. Yeah, I just think that's, topic. Most commonly on long FRQs, yes, you're going to. One of them is going to be like experimental design, like this one, right? It's going to be something like this, where it's asking you about dependent variables and that sort of thing. One of them's going to be that, and the other one's going to be a graph every time. But I don't know. Like this one's about cellular respiration. This one's about cell signaling. I think it's a crapshoot. I think you could probably do all the guessing in the world and not get it right. But you could definitely see like a giant pedigree or something here and them ask you a bunch of questions about that. So, all right. So let's talk about fermentation. Glucose is broken down in the presence of oxygen to make ATP. That is cellular respiration. What if oxygen is not there? Well, that's when fermentation occurs. However, I want to go here. What is this process called right here? This process is called glycolysis, breaking down glucose to make 2-pyruvate. All organisms, all living things do glycolysis. So that's important. Doesn't matter if there's oxygen. Doesn't matter what's going on. Notice that decision is not made till after this. And so make sure that you understand glucose to pyruvate, everything's doing that. And that's step number 1 in cellular respiration, by the way. Doesn't require oxygen. Where does it happen? Happens in the cytoplasm. And so for some organisms, well, that's all they have, right? They don't have a nucleus. They don't have a mitochondria. They don't have any of that stuff. It doesn't mean they can't do cellular respiration, but they are all doing glycolysis. Glycolysis is a conserved process, meaning that's all organisms do it. All right, so let's say there's no oxygen in the cell. Well, there's no oxygen, and so we're going to go through fermentation. What's the purpose of fermentation? Well, let's look at it. All right, so let's, first of all, before we look at my little definition up here, here's glycolysis, glucose to pyruvate. And notice what's made here. So some ATP is made, and there's some NADH. NADH is an electron carrier. This electron carrier is going to go to ETC to make ATP. Well, if there's no oxygen, there's no ATP being made, right? And so where is this NADH going to go to drop off its electrons? It's not. because oxygen is the final electron acceptor in cellular respiration. So if there's no oxygen, NADH can't drop its electrons off. It's like filling up a bucket of water and standing there with no place to drop it off, right? You can't go get another bucket of water until you drop that water off, and you're kind of stuck. You need a place to drop your water. Well, fermentation is the place to drop that water. NADH is going to go drop off its electrons here in this reaction. This is called NAD plus regeneration. If I can find my cursor. Yeah, regeneration. It is making more NAD plus. Why is it doing that? Because NAD plus is necessary to make NADH, which is necessary to do glucose to pyruvate. And so fermentation is happening here, this process of converting pyruvate to lactate. The purpose of it is so that this process right here, glycolysis, can continue. So how can we define that? Well, if there's no oxygen, so glycolysis can continue. Frees up electron carriers, creates NAD plus from NADH. And so the purpose of fermentation is to free up electron carriers so that glycolysis can continue. This is animals, production of lactate or lactic acid. I don't have the ethanol one, but it's just ethanol and CO2 is being made. That's what this is, ethanol and CO2. It's like bread or beer, something like that, right? But if there is oxygen, then oxygen is going to be the final electron acceptor. This NADH is not going to go through fermentation. Instead, it's going to go into the mitochondria, and it's going to drop off its electrons at cellular respiration, or at the ATC. Speaking of that, let's talk about the steps of cellular respiration. Where's my picture? It's on this one. This is my favorite picture. This is another one of those pictures that I run off for all my students so they can have this thing and it shows you, what you're looking at or whatever. Because it's one of those things that I tell them. It's like, hey, if you have this, then you can know the whole thing. So here's glycolysis again, glucose to pyruvate. There's some ATP that's being made here, right, but not very much. And if there's oxygen, this pyruvate's going to go into the Krebs cycle. This is what we call pyruvate oxidation. Where is it? It was on this right here. Pyruvate oxidation. I don't really like that they made a special thing for this. It's fine. But basically, what is pyruvate oxidation? The pyruvate's got 3 carbons. It's going to lose one of those carbons as it's oxidized. Anytime that carbon's given off, it's given off as a CO2, which is a byproduct of this reaction. Some NADH is going to be made, and then you have 2 carbons left, and this enzyme called coenzyme A is going to pick up those 2 carbons, and it's going to put them on the merry-go-round. That merry-go-round is called the Krebs cycle. Here it is right here. And so here you can go back up here. You see pyruvate is there. Actually, you know what? We're not going to do this one. Let's do this one simpler. So there's pyruvate. It's got its 3 carbons. It's going to lose 1 CO2. NADH is going to be made. Here comes coenzyme A. It's going to say, hey, I'll help you get on the merry-go-round. It's going to put it on the merry-go-round. And those 2 carbons get on the merry-go-round. They're going to jump off the merry-go-round as CO2. It's going to make some NADH, FADH2, and ATP. And so I know I'm talking really fast, but I'm just trying to going through the details fast so that I can come back and go over the overview slower. So that pyruvate, if there's oxygen, is going to enter into the mitochondria and it's going to go through the Krebs cycle. Well, let's look at this picture. What's the purpose of the Krebs cycle? There's a little bit of ATP made, but it's not very much. The main purpose of the Krebs cycle is to break down pyruvate to make electron carriers. Yes, lactate is found in muscle cells. And ethanol is in yeast and some bacteria too. Also can produce ethanol as well. Good question. So the purpose of Krebs cycle, make electron carriers, right? Make some ATP, but the main purpose of it is to make electron carriers. And here's some electron carriers too. Where are the electron carriers going? Well, they're going to the electron transport chain. What's the purpose of the electron transport chain? Electron transport chain is using these electron carriers to make ATP. This is what we were talking about earlier in the stream. Is we were zoomed in on this picture, and we were talking about how this particular version of the ETC is using the energy that's in these electron carriers to power this process of making a proton gradient, which is called chemiosmosis, in order so that this ATP synthase can then use that gradient to make ATP, right? And so the purpose, going back to this picture of the ETC, is to make ATP using the electron carriers. And you will see this term, oxidative phosphorylation, associated with this. So let's think about this word, right? Oxidative, what does that sound like? It sounds like oxygen, right? So oxygen is necessary for this particular reaction to take place. Phosphorylation, what does that sound like? Sounds like phosphate, right? It's because a phosphate is being added to ADP. Let's see if I can find it. Bloop, 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 bloop. I bet I have it on this one, maybe. Perfect. Here we go. All right, so it's when a phosphate's being added to ADP. Here's the phosphate. Oxidative phosphorylation. It's happening in the presence of oxygen, right? And then you probably saw this other thing here where it was called substrate level phosphorylation. Where'd my picture go? Substrate level phosphorylation. What does substrate make you think of? Enzymes, right? Because this is all powered by enzymes. And you can see that on this picture here. Not this picture, wherever it is. It's on this one. Like this Krebs, it's crazy. See all the ACEs, so many enzymes involved in that. So yeah, that's steps of cellular respiration. Seriously, that's a lot of weeds and it sounds really complicated. But if you had this picture on the exam, you wouldn't need anything else. They don't want you to have this picture because it would give you too much info. That's how good it is. I'm like really convinced on this picture. Same thing for photosynthesis. There's a big version of this for photosynthesis. It's this one. If you had this picture on the exam, they don't want you to have it because it gives you too much information. It's just everything you need to know is right here. Good questions. Anything else before we call it an evening? Or you know what? I can be here a while. I don't really have anything to do except for go to sleep. And I'm kind of not feeling very tired. So, yeah. Let me know. Anything else you want me to go over? Oh, how are the bonds broke during crossing over in prophase 1? That's a really good question. I guess yeah. Jeez. I don't even know. You talking about like the covalent bonds that are holding the DNA together. I don't know. It's definitely beyond the scope of this class. You will not need to know that. If you do need to know it, it will be given to you. But my guess is that there's some sort of enzyme involved with that. Because it would definitely need to be a controlled process, because if it's not a controlled process, it could happen, it could be crazy. And crazy is not good. Cells don't really like crazy. So that's not a question that I know the direct answer to. Never been asked that question. So yeah, I'll look it up. Good question. Anything else? Bonds broken. Golly, that's bad. The spelling. Yes, I could definitely go over cell signaling. There is a protein called SPO11 that does those breaks. There you go. It's just some sort of enzyme that you definitely do not need to know. All right, so let's talk about signaling. You know what? In order to talk about signaling, I am going to use this practice as exam that we were looking at earlier, because I think there's a good picture on this that kind of talks about signaling. Here it is. Perfect. Actually, no, this is not good. It's not as good as I thought it was. Okay, so let's just start here. Cell signaling, what is it? It's just communication. That's all it is. It's all in a practice FRQ. Interesting. Yeah, that would be the kind of thing that they would like literally give you. Was it that one? No, that's not the same one. Yeah, I would give you that information. It's not going to like ask you to like come up with that. So cell signaling is communication between cells, and communication can happen when cells are touching one another, like these two cells are touching one another, or it can happen with more like distance relationship, right? So like a cell can talk to all the cells in a general area, like we would call that like local communication, or like distance communication. So here's like local regulators, or it could be like long distance, where one cell puts a hormone into the blood and then another cell on the other side of the body receives that information, right? So I have some examples there. The general signaling pathway, I use a telephone in my own class to talk about that, but we're just going to skip that. And we're going to have to use these 3 steps. It's called the signal transduction pathway, and it's 3 steps: reception, transduction, response. And all of these are involved in the process. Reception is the literal reception of the signal molecule. The signal molecule is usually generically called a ligand, Ligand. Shouldn't try to spell, I'm dyslexic. So a ligand is a signal molecule, and it's going to be received by some kind of protein. Typically, it doesn't have to be received on the cell membrane. It could be received inside the cell, which I might have a picture of that. Yeah, here it is. So like, here's a ligand being received on the outside of the cell. Here's one being received on the inside of the cell. Typically has to do with the chemical makeup of that particular signal molecule, whether or not it can pass through the cell membrane. And so that is reception, right? This is going to trigger some sort of response from this protein. We know that protein's shape determines their function. And so what is going to happen to this protein when this little popcorn kernel binds with it? It's going to change shape. When it changes shape, there's going to be some sort of energy transfer there, right? And so what you see here is this transduction step. This is literally like a relay where this bean turns on this bean, which turns on this bean. Think of it as like a pathway, literally a pathway, right? I always use like, so in my classroom, my where I usually stand is in one end of the room, and then there's the classroom, and then there's a lab, and then there's the door, right? There's not 2 doors to my classroom because that would have made too much sense. So they only put one all the way on the other end of the room. And so like a student knocks on the door and has a note for me. Well, I ask a student to go get that note, and then that student goes and gets the note and brings it to me. That would be like a transduction, right? The door is where that student was received, and so that'd be reception. The note is the actual signal molecule, but that's going to cause this whole cascade of things. Maybe 2 or 3 students get involved in the relay process and make a game out of it. I don't know, it doesn't matter. And that whole relay process is transduction. The response is me reading the note and doing something about it, right? And so this response is going to be some kind of cellular process, activation of a cellular process. How do cells activate cellular processes? Typically from the production of a protein. And so this is going to go into the nucleus. It's going to cause transcription of some sort of protein translation, or transcription of the DNA into RNA and RNA into protein. Good. What does a second messenger do? That's a really good question. So there's reception. Ligand binding domain. I think here, just make sure that you understand that each receptor is specifically fit for a particular ligand. They're not general. G protein. We're not going to talk about G proteins. Here we go. Secondary messengers. Perfect. And so let's just look at this picture. Let's say that we have no knowledge. We've never heard of this before. Good question. How's the signal amplified? We'll talk about that. So we'll just say we've never heard anything about any of this stuff before, right? Like we just come in and then we see a bunch of like beans on the page, and we're like, okay, what's going on? Just my simple like knowledge. I see this messenger. It's called a signal molecule, right? And it's turning this on, which is turning this on, which is turning this on, which is doing this, which is doing this, right? So what is the secondary messenger here? What role is it playing? It's just a step in the pathway, right? Now, cyclic AMP, which is CAMP, you can call it CAMP or you can call it cyclic. That C is cyclic AMP. We know ATP, we know ADP, right? Now we have AMP, adenosine monophosphate. Through a lot of proteins being phosphorylated. Absolutely. See this GTP, see this ATP. Every one of these little processes has to do with phosphorylation of a protein. What's going to happen when you stick a phosphate on this G protein? It's going to change shape. What does the shape do? It changes its function, right? So yeah, absolutely. Phosphorylation is occurring on each step. Same for this cyclic AMP. It's going to have a phosphate added to it. It's going to become something different. It's going to turn on this kinase. When you see the word kinase, that has to do with turning something on. So that protein kinase is going to go turn something on, right? So this is very general, right? G protein pathway. Sometimes you'll see it used. It's one of the most common pathways in eukaryotic cells. I think there's some in prokaryotic cells too. So very simple pathway. So how can that signal be amplified? It's a perfect picture right here. I love this. Call it signal cascade. So here's the ligand. How many signal molecules is there? Right. This is the important This is the purpose of cascades. Because how many signal molecules do I need in order to elicit how many ever green peas that is? It's a lot, right? Or it's a lot of responses for one signal molecule. So the purpose of a cascade is to use less molecules in order to elicit more response. Well, how does that happen? Well, the transduction part of it. So this is reception. Transduction is going to turn on these three beans. These beans are going to turn on three beans each, and they're each going to turn on two. And so then it's like I would say it's like a phone tree, but you guys don't know what a phone tree is. Back in the day when you get a call from the principal and he would say, hey, call these three people, and tell them that there's no school. And then those three people would call three more people, and it would cause this cascade, right? It's the same kind of idea. It amplifies the signal. It amplifies the response without using very much of the signal. And I think that is the purpose of that. And yeah, this shows that modification that you were talking about with the phosphorylation as well. Anytime you add a phosphate to something, it's going to change the shape of that protein. It's going to change its function. And the same thing is going to happen when you take the phosphate off, right? If you take the phosphate off, it's going to reset that protein, so to speak. Dephosphorylating it. Good. Secondary messengers, ligand-gated channels. This is very simple, right? You got your ligand opens the gate, so all the green dots can go in. Good. So the thing about cell signaling is that it's going to be very, very specific, right? It's thank you for the compliment. I appreciate that. It's going to be very specific. Go back to this picture here that we were looking at. It's going to have some sort of very specific kind of setup here, and no one's ever heard of these. I mean, maybe somebody's heard of these, like one of your teachers or something, but more than likely you haven't. The purpose of this is not to know about the PHO4 genes and all this stuff. The purpose of this is to be able to look at this and be able to interpret what you're seeing, right? And so if we just look at this, and we know that PHO81 is some sort of activator or something, right? And I can go up here in the prompt and I can find out about it, right? But PHO4 is being phosphorylated here, right? Because ATP is going to ADP. And when that happens, these genes aren't being transcribed, right? Yeah, this question was actually from the 2023 exam. So this is from last year's exam. And I actually made videos of myself answering all these questions too. This video is actually one of my most popular one on my channel right now. This me answering this question. Notice if that PH4 is not phosphorylated, then these genes are being transcribed. And so when this PHO or PHO4 is phosphorylated, it's essentially controlling transcription. So this is a form of gene regulation. And so this is crossover, right? This is unit 4, cell signaling, but this is also unit 6, gene regulation, kind of all mashing up together. And so make sure that you don't get lost in that either. But yeah, very simple. Let's go back to cell communication, see if there's anything we left off. I don't think so. A lot of these parts are very similar to one another. There are things that can change that, obviously, very straightforward. We talked about cell cycle, but cell cycle is very closely related to signaling because the cell is going to receive signals from the outside. in order to either elicit or prohibit cell division. And when a cell's unable to control division, that's what cancer is, right? When the cell's unable to regulate its cell processes. So, good. Any other questions? Good questions so far. Very, very helpful. Appreciate you guys' respect in the chat as well. Very, very much. Your teachers would be proud of you for being respectful. I have seen some other teachers' streams that have not been very, very respectable. So this has been good. Not because those teachers aren't respectable, but because the students on them aren't. Teachers are trying to do a good job for you guys, but good. Anything else? Any other specific questions that just burning a hole in your mind? Or is it's 9:30 in the Central Time Zone? I don't know where all you guys are from, but here it's starting to get pretty late. Maybe time for bed. We need to know what happens at each checkpoint. Yeah, you might need to know what happens at each checkpoint. So I would have a basic understanding of it, though, right? So, okay, like, we're trying to get 5s. I understand, but you can't get a 5 if you're tired. You can't do it. At least I couldn't. So what? Yeah. So if you know what's going on in G1, right? Growth, all that kind of thing, nutrients, and so the G1 checkpoint is basically checking for those things, right? One of the things I tell my students is the G1 checkpoint is oftentimes called the point of no return, because if a cell makes it through the G1 checkpoint, that cell is probably going to divide. Why is that? Well, the next phase is S phase, and during S phase, DNA replicates, and there's no reason to have 2 copies of the DNA unless you're going to divide the cell. And so some cells will actually stay in G1 for the rest of their lives, just chilling, doing the G1 thing. All right, and G2 checkpoint is, okay, DNA just replicated, right? And what comes right after G2? Mitosis. And so G2 is going to be checking to make sure everything's ready to do mitosis, right? Yeah, I can do that. Functions of organelles, I can do that. And so the M checkpoint, very specific. The M checkpoint is to make sure that each one of the sister chromatids is attached to a spindle fiber before they are disconnecting. It's kind of like, imagine you're like trying to pull a boat up, like someone's bringing their boat in. If you do fishing or boating, you know what this is. If you don't, probably completely lost. But you want to check and make sure the boat is like attached and connected before you pull the truck forward, right? And so it's the same. It's making sure every chromosome is exactly hooked up before the cell splits so that there's no mistakes. Let's look at cell organelles. Where is they? Can't even talk. All right, so here we go. Cell structure and function. Subcellular components. There's a blank there. Ribosome. It's the purpose of ribosome. It's a site of protein synthesis, right? ER is the there's 2 kinds of ER, so you can define them separately, but we'll just look at them. There's smooth ER. A lot of functions for smooth ER, but I want to say that the course description actually like explicitly states that you don't need to know all the functions of like specialized smooth ER. If you know this is where like membrane lipids are made and like a detoxification, smooth ER is good. And rough ER is where ribosomes are going to be. And so if ribosomes are there, what is the rough ER going to be associated with? Proteins, right? Associated with proteins. So they're going to go in there. It's like a changing room for the proteins that go in there and like fold up and get ready. Where are they going? Well, they're going somewhere. They're going to the Golgi. What's the purpose of the Golgi? The purpose of the Golgi is to modify proteins for shipment out of the cell. Speaking of that, if a protein is made by a ribosome that is on the rough ER, that protein is leaving the cell. All right. If a protein is made by a ribosome that's floating around out here random, it's going to stay in the cell. But these proteins that are made in here, they're getting ready to leave because they're going to be put in a little bag. I don't have a picture of that little bag. Here's the little bag. It's these vesicles. It's these other vesicles up here, incoming transport vesicle. Where's it coming from? It's coming from the ER. It's a little protein in there. It's full of like hopes and dreams. and it's going to come in here and it's going to bind with this Golgi. The purpose of the Golgi is to modify the protein. Why is it doing that? Well, the protein has a destination, and all the cells are just waiting with bated breath to see this protein, wondering if it's for it. It's like, is that the mailman? Does he bring me anything? And the little modification on that protein is going to basically tell another cell, well, that's mine, or no, that's not mine. Right, so that's what the Golgi's doing. So the Golgi does all that in this series of little pancakes right here, and then it's going to make another vesicle, and it's secretory vesicle. Think the word secrete, which is to get rid of that protein. That protein is then going to leave the cell. Sometimes you'll see the rough ER and the Golgi and all those vesicles called the endomembrane system, and that's their purpose, is to package and ship proteins out of the cell. Mitochondria, the purpose of the mitochondria is to produce ATP, break down sugars to produce ATP. ATP is cell's energy currency, adenosine triphosphate. There's that. Lysosome. Lysosome is it's like a cellular stomach, kind of. It has a really low pH inside, and it will break down things. And so let's zoom in here. And so this cell's going to take in some sort of bit of like mushy cereal or something, and this is why this is called a food vacuole. And then this food vacuole is going to combine with this lysosome, and now this whole thing is a lysosome, right? Because the whole inside of this is going to have a lower pH. It's going to break down that mushy cereal and to make it into something that you can actually use. And so that's what lysosomes do. But it can do that with a virus. It can do that with bacteria. Anytime something needs broken down. In fact, if the cell needs broken down, like if the cell is just done, or maybe there's something wrong with the cell and it wants to do apoptosis, like programmed cell death, the lysosomes will just break themselves open, and the whole inside of the cell becomes a lysosome and eats itself. True story. Vacuole storage. Vacuoles are going to be associated with plant cells primarily, to store water, but there are other cells that have vacuoles as well. Large central vacuole. Chloroplast, photosynthesis. That's not all of them. Maybe it was. This picture's always so weird to me. I think that was it, actually. Specific questions? Anything specifically about any sort of cell part or function that you're curious about? This picture. My students, apparently when I describe this to my students, this endomembrane, or this endosymbiosis, I talked about this being a Cheeto and this being pickles. And all of my students have Cheetos and pickles written in their notes. And so I thought that was really, really funny. Really funny. Yep, good. Questions, other thoughts or questions before we sign off for the evening? There's still quite a few in here, so I want to make sure that everyone's has their questions answered and feels like they're prepared for the test. Or if there's a question about the test itself, lots of questions like that have come up tonight. And so please feel free and ask those questions as well, because I have a test. I have a sample FRQ right here in front of me anyway. I don't have a sample test because we don't get those anymore. But yeah. Any other questions? So it's just kind of a selfish plug for myself while you're all listening. So Thursday you've taken the test and you're like, what can I do on Friday to really celebrate? On Friday you can go over to twitch.tv and you can find Chip Mafia and you can watch me play Fortnite with my friends. We're bad and it's funny, so you should go do that. Just go follow me on Twitch. What's up, Boxer Dog? Thanks for joining. Can I go over transcription, translation? Man, I would love to. And go over water potential? Yeah, I can do that too. Let's do it. All right, so transcription, translation. Let's talk about this picture's perfect for that, actually. So first of all, big ideas for transcription translation is barely taught ecology. Yeah, I've heard that a lot. What's the bare minimum I should know? Ecology is Unit 8's a really easy unit, actually. It's almost like Unit 7, Part 2. And so as long as you understand that organisms change according to their environment, you're going to be fine. And if they don't change, they die. Yeah, you'll be good. I actually made a video, Unit 8, and it's like 20 minutes long, that I reviewed all the content for the unit as well. It's you could watch it on double speed because I talk slow. All right, transcription is going from DNA to RNA. That does not mean that you're converting DNA to RNA. That means that you're reading the DNA, and from that DNA, you're creating a complementary RNA. Yeah, ecology, not hard. Most of it you had in like 7th grade. You'll be fine. Translation then is taking that RNA out in the ribosome, or out in the cytoplasm on a ribosome, or the rough ER for that matter, and you're translating it to make a protein. All right, so that is the basic. Common mistake. Students get these things mixed up, right? This whole thing is called protein synthesis. All right, so let's see. Wow. Okay, this is actually a really good picture. Man. All right. So here's my DNA. Here's transcription. Transcription is going to use the template strand, or the non-coding strand, which is, I know, horribly confusing, in order to, because there's 2 strands of DNA, right? But only one of them is going to be read. And so it's going to use that to make a complement. And then that is going to be used in groups of 3. Those groups of 3 are called codons in order to make the polypeptide. And how do you know what those are code for? Well, you're going to be given a codon chart, which is right here. And if you need a codon chart, you will be given that, by the way. That was the general overview of transcription and translation. It's typically going to be that simple, right? I would say that if you know that simple idea, then you'd be able to answer anything more complex than that. I think one of the things that we do a lot of times as teachers is we go way too deep, and we like lose the students up at the surface sometimes. And I try to keep everything pretty surface. You do not need to memorize that chart. No, that'd be horrible. Probably the only thing that I would remember is that AUG is the start codon. But AUG is also methionine. Individual steps of the Krebs cycle? No. You do not need to know the individual steps of the Krebs cycle. You just need to know that the Krebs cycle makes electron carriers. It's good. AP Bow in a few hours, yeah. Join the crowd. Let's see. What was the question that I don't want to make sure I missed? Oh, water potential. Yeah, let's do that. Okay. I think that's here. No, it's not. It's here. Water potential. Here we go. Okay. You get to run a game of Fortnite with me? Yeah, I'll do that. If you can prove that that you made a 5 on the exam, I'll play Fortnite with you. That's good times. Chip Mafia is my name on Fortnite too. So, I mean, I play with my friends, and we're all like middle-aged dudes that are bad at the game. Our whole purpose is to have fun, and I have lots of silly clips of me playing on my TikTok and some other things. So, yeah, we just have fun. The whole purpose is fun. So, come out and join, laugh with us, laugh at us. We don't care. All right, so water potential. Let's talk about water potential. First of all, let's make sure we understand a couple terms. Solvent, we know what solvents are. We're not going to do that. Tonicity. So here's the terms that we need to know. Okay, hypotonic, hypertonic. These terms have to do, first of all, they're comparative terms, right? I'm actually going to not use these pictures. I'm going to draw my own thing over here. So here's a picture of All right. Thanks for stopping by, Boxer Dog. Thank you. See you later. Good luck on the exam. So here's a cell, right? And this cell's got a bunch of galaxy dots in it. And then outside, there's not very many galaxy dots, all right? And so a third grader could look at this and tell you that the inside of the cell is more concentrated than the outside of the cell. And so let's step it up a little bit, right? Which side of the cell has more solute? Well, the inside of the cell has more solute than the outside of the cell. Okay, good. Which side has more water? That's the real question here. Well, the outside has more water, right? Good. And so let's say that this membrane does not allow the passage of galaxy dots, right? But it does allow the passage of water. And so which way is water going to go? Well, it's going to go from a place where there's more water, which is outside, to where there's less water. And so water is going to want to go into the cell. All right. So what's a rule that I can come up with that doesn't require me to think about galaxy dots? So a good rule is, well, if the inside of the cell has more solute, that inside is hypertonic. These words hypertonic has to do with, and hypotonic have to do with the amount of solute. And so if the inside of the cell is hypertonic, then the outside of the cell is hypotonic automatically, right? And so inside is hypertonic, outside is hypotonic. Water always moves from hypo to hyper, right? So that's, now that we have that, we're going to move on. So let's do some of those equations and things. This is one of those things, by the way, this water potential, much is made about it. It's not hard. And so using that same information, talking about water potential is just water's tendency to move. We just learned that water goes from hypo to hyper, right? So if we look at this particular picture, let's look at it without looking at the numbers. And the solute is gray dots and the water is white dots. Well, which side of this membrane is hypotonic? This is hypotonic right here, right? And the inside is hypertonic because it's got more solute. So where's water going to want to go? It's going to go from left to right, or hypo to hyper. Well, how can we translate that into these numbers? Well, what number's higher, 0 or negative 200? Oh, number line, right? 0 is higher. Water always goes from a high water potential to a low water potential, right? It's a good rule of thumb. I've seen students miss a question like this because they forget that negative 200 is lower than 0, so don't do that. And so here's the equation. How do you get water potential? Well, it's solute potential plus pressure potential. A couple of things you need to know. Pressure potential is always going to be given. You're not going to have to solve for it. Solute potential, you will have to solve for it. All right, let's find it. Here's the equation for solute potential. There's a couple things. Here, let me get my highlighter out. Molar concentration, you don't have to solve for moles in this class, so it's going to be given. R is always given. Here it is on the formula sheet. Temperature is probably going to be given to you in Celsius because they want you to do this little thing right here, right? Which I also believe is on the formula sheet. Let's double check to make sure that's correct. Here's the formula sheet. And here's our water potential business over here. Yep, and there's our conversion. So this is what you're going to be given on the actual exam. One other thing. Oh, this is just for my class. Yeah, sorry. Go over gene expression. Yeah, I can go over gene expression too. Yeah, and this is just like my slides. It's nothing special about any of this. So here's that formula sheet, right? What was I doing? Okay, yeah. So here we go. So really the only part of this equation that you're going to have to worry about is this ionization constant. And what is ionization constant? Ionization constant is the number of pieces that are made when you dissolve this chemical in water. It's going to be 1 or 2. That's what I tell my students. If it's a sugar, it's 1. If it's anything else, it's probably 2. If it's an acid or a base or a salt, it's going to be 2. If it's a sugar, it's going to be 1. I've never seen it be 3. It's not going to be 0. And the thing that students miss the most is not the ionization constant. This is what they miss the most. They forget to multiply that number times -1. So do not forget to make this number negative. Solute potential is either going to be 0 or it's going to be negative. It's never going to be positive. And so make sure that you understand that. Yes, I can do that, Dubs Factory. I can definitely do that for sure. So let's see. That was water potential. Oh, yeah. And then you add those numbers together, right? So you're going to have pressure and solute. That's going to be 1. You're going to have that on the other side. You add it together. And then which one's higher? That tells you which way water goes. Hopefully that was helpful. I know I went really fast. Individual steps. Calculations. I think I did that. Gene expression. All right, let's talk about gene expression. Gene expression is really simple. This is gene expression. A gene is a section of DNA, right? And a section of DNA. Thank you, grape juice. Appreciate that. So gene is a section of DNA, and it's A's, T's, C's, and G's, and that gene is expressed when the protein is made, right? And so if you're regulating gene expression, which is something, what is this? Gene expression and cell specialization, regulation, gene expression. All of this concept has to do with the regulation of the production of that protein, 100%, right? And so if I'm regulating gene expression, then I am regulating the production of a protein. What does gene expression have to do with cell specialization? Well, if a cell is specialized, let's say a muscle cell is making things, specifically is making proteins that a nerve cell or that a fat cell is not making, right? And so because it's making this particular set of proteins, it is different than a cell that's making a different set of proteins. And so that is all that's meant by gene expression. That's it. This is if a protein is being made. Good question. Let's go back to this Unit 3. Answered a lot of test-taking tips. Keep your answers short and don't spend a whole lot of time on a particular question. Just kind of move along. Particularly for a science AP exam, just answer the question. Don't explain yourself. Unless it asks you to. If it asks you to justify your response, then please justify your response. Otherwise, don't. Just keep it simple. Don't write too much. That's probably the number one mistake I see my students doing is they're spending too much time writing things that I'm not even going to read because it's not part of the answer. Okay, cellular respiration. And no, wait, you specifically asked Krebs electron transport chain stuff. All right, let's talk about that then. Good. It's this picture right here that's so good. This picture. Seriously, go out and Google cellular respiration and find a picture that looks like this because it's like the secret, right? Because it tells you what's the arrows, you follow the arrows, everything's perfect, right? These are the three steps of cellular respiration: glycolysis, Krebs cycle, etc. Glycolysis happening in the cytoplasm, right? And thing about glycolysis, doesn't require oxygen, and it happens in all organisms. And just look at this picture. What is glycolysis making? It's making pyruvate, right? Where's pyruvate going? It's going to the Krebs cycle. But it's only going to go to the Krebs cycle if there's oxygen. If there's no oxygen, it's not going to the Krebs cycle. It's going to do something else, which is right down here. It's going to go through fermentation, which is this whole other thing. Right, so so the purpose of glycolysis, make pyruvate. There's a little bit of ATP made. There's some NADH made, but the main purpose is to make pyruvate. Where's pyruvate going? Krebs cycle. What's the purpose of the Krebs cycle? The purpose of the Krebs cycle is to make electron carriers, right? Because electrons are carried by NADH and FADH2. And so that's the purpose of it. There's some ATP made, but the main purpose of electron carriers or the main purpose of the Krebs is to make electron carriers. Notice where all the electron carriers are. It doesn't matter where they're made. Look where they're going. They're going to the ETC. What's going on in the ETC? Well, the purpose of the ETC is, in the presence of oxygen, to use the electron carriers to make a concentration gradient in order to make ATP. What does that look like? Let's go find it. Here it is right here. All right, and so there's NADH, FADH2. They're dropping their electrons off. They're carrying them here, right? They're dropping them off, and they're building up this gradient. This gradient's going to be used to make ATP. This has to happen in the presence of oxygen. There's oxygen. It's the final electron acceptor. One of the cool things about this is, and I tell my students this all the time, that cellular respiration and photosynthesis are like antithesis of one another, like opposites. So like literally the first thing that happens in cellular respiration, or in photosynthesis, excuse me, is that, look at this. I mean, isn't this crazy? H2O is broken down, oxygen's given off, right? Notice what's the last thing that happens in cellular respiration. Oxygen's being used, and electrons are being added to it to make water, and so it's the exact opposite, right? And so if you think about them in that way, it's a lot more complete system. I think that's really helpful. The purpose here is to make ATP. Big idea. Can I go over meiosis? After this, I don't get the different phases of meiosis. How it can? Perfect. We can do that. Unit 5. Meiosis. You know what? I did this earlier. I'm just gonna draw it up here because I can talk about it better. All right, so we're just gonna use 2 pairs. This is gonna get a little tedious, but it's worth it. And so I've got this pair. Actually, you know what? And then hold on 1 second. There's that. And this other one's going to be purple and yellow. And so this particular chromosome has, or this particular cell, this is a cell, where I'm not going to draw a circle around it, but you can just imagine there's a cell. This particular cell has 2 pairs of chromosomes, or you would say that 2n equals 4, right? Is the diploid number of this. 2n equals 4. Well, after meiosis, or after interphase, what's going to happen to interphase? What happens to DNA? Well, DNA replicates, right? So this is when you're going to have this kind of shape of chromosome that you're probably used to seeing when you think of a chromosome. Where'd my red go? Seriously? Do I have to come up with my own red? Let's hope for the best. Well, it's not that great. Okay. And then purple, and then yellow, right? And so now I still have 2n equals 4, but each one of those are duplicated, right? And so a couple terms here that we need to get. First of all, these 2 pieces are sister chromatids, right? They're identical to one another. These 2 pieces are identical to one another. Sorry, you have a question. Why are they? Okay, yeah, good. So usually on the pictures you're going to see the X's already because it's assuming that this particular thing has already happened, interface. And so let's go here. Here we go, right here. See this right here? You got two pairs, and this is the straight lines. And then once interface happens, that's showing the replication of DNA. That's what that's showing, right? And so, so that's what I showed here. And so each one of these are called sister chromatids. Yes, I can go over the different kinds of macromolecules for sure. All right, sister chromatids. Red and blue are homologous pairs, right? Red and blue are homologous pairs. I got red from my mom, and I got blue from my dad, and that's a pair. They're homologous pairs. They're not identical, but they do have the same genes on them, kind of like my shoes. Left shoe and right shoe are not identical, but they're the same shoe, right? It's normally just lines, not X's. It's only X's after they've replicated. Why do they need to replicate? Well, I'm getting ready to show you. If they don't replicate, you don't have enough for the cells at the end, right? And so the first thing that happens, meiosis 1, is homologous pairs separate. And so when homologous pairs separate, red is going to go here, or blue is going to go here, red is going to go here, right? And so now they're no longer diploid cells because they've only got half. Now here's another teaching moment right here. Where's purple go? Does purple go with blue or does purple go with red? It doesn't matter. Could go with blue, could go with red. And that's that principle of independent assortment, right? And there's, we could go into a lot of depth here, but we don't have a lot of time. If I don't want to confuse you all. And so we're going to say that purple goes over here and yellow goes here, right? And so that is meiosis 1. Well, what happens during meiosis 2? In meiosis 2, sister chromatids separate. And so if sister chromatids separate, then this, when are you ending? I don't really have an end time. I mean, I'm eventually going to go to sleep because I'm old. But and you guys need to go to bed too, but because you have a test tomorrow. It's pretty big. And so sister chromatids separate, and it's going to look like this. And then these are like haploid cells. Remember when we said 2n equals 4, and our original cell looked like this, right? Not crossed over, but looked like that. Now we have 1n equals 2, right? And if purple had went with red, then you might have this combination. And so it just shows you that there's different combinations in cellular in sexual reproduction. Macromolecules. Let's talk about them. There you go. There's a fun picture. I don't know why I have that picture. It's literally the first slide of the whole. No, it's 1.2. Never mind. Let's see. Introduction to biological macromolecules. Here we go. Okay, here we go. There's 4. There's nucleic acids, proteins, carbs, and lipids. How do you know what you're looking at? It's a couple rules of thumb. Nucleic acids are easy because they always look like this every single time. They're going to have a phosphate, this 5-carbon sugar, and a nitrogenous base. It's going to have 1 or 2 rings. They're always going to look the same. They always have phosphorus in them. If you see a P, it is a nucleic acid, 100% chance. If you see an N, it could be 1 of 2 things, but the P is always nucleic acid. So the homologous chromosomes align during metaphase. Yeah, they do. I actually don't spend a whole lot of time talking about metaphase and telophase and all that stuff with meiosis because I think that just the concepts of homologous pair separating meiosis 1 and sister chromatids separating meiosis 2 are the primary concern. Some of the other stuff is not that important. I feel like you can get lost in those details and miss the big picture. It's just kind of the way that I think about a lot of stuff. So, good. Nucleotide, phosphate, pentose sugar, nitrogenous base. How do you know it's not a protein? Well, first of all, a protein doesn't have P in it, right? So here's a protein. This is an amino acid. Amino acid has an N in it. It's also going to have an R group. It's not going to say R. It's probably just going to be like random. These have R's in them too. There's 20 different amino acids. It could be anything. It might have an R in it. The reason you know it's a protein is because it has an N in it and it doesn't have a P. Very simple, but you probably knew that a nucleic acid or nucleotide is phosphate, 5-carbon sugar, nitrogenous base. And here's the amino acids. So this shows you the R groups. I actually saw a question on a practice exam that said, "What is a carbohydrate composed? What atoms is a carbohydrate composed of?" So that's not a bad question. Proteins are composed of lots of stuff, but never phosphate and always nitrogen. Sometimes sulfur. Where's sulfur? cysteine, there it is. And there's one more, I guess it's up here, methionine. Yeah, there it is. So there's sometimes sulfur. And carbohydrates, always three things, C, O, and H, and the C's and the O's are equal every time. The amount of C and the amount of oxygen is always equal. So in the glucose, it's C6, H12O6. The H's may not be twice the number of C's, may not be, sometimes is, but not necessarily. But the C's and O's are always equal. And a lipid's also going to have CH and O, but there's very few O's compared to the C's. So notice. So it's like a couple of O's. And the lipids are typically in these longer chains, right? Because they're nonpolar. Man, I've been at this for a couple hours now. Still have quite a few in here as well. No, that's a good question. The carbon may change. So you can have a carbon with you can have a carbohydrate with 10 carbons in it, but it's also going to have 10 oxygens every time. The way that you know you're looking at a carbohydrate is that the carbons and the oxygens are equal. So as far as macromolecules are concerned, you start getting inorganic chemistry, then you're drifting out of my zone there. I do have a good friend. If you're planning on taking AP Chemistry, you should go ahead and go sub to his channel. His name's Jeremy Krug, Mr. Krug, AP Chem. Go and sub to his channel. You will not be upset. He's the best AP Chem teacher out there. So teaches next door to me. It's quite a privilege to have him next door. Let's see, what else? Lipids, talked about that. There's special kinds of lipids. So, blip, blip, blip. Structure and function, very important. Yeah, I'm feeling myself getting like less with it mentally. So I'm probably going to need to sign off here pretty quick. So real quick, before I do sign off, I made lots of like YouTube shorts also, if you're familiar with that. I know that you guys aren't because you use TikTok, but YouTube shorts, if you go to my channel, there's a whole playlist of them, and it's literally like 20 seconds through each little section, sometimes more. And so you can just sit, it's almost like little flashcard videos. And so kind of review, strongly encourage you to go watch those while you're going to sleep. Phospholipid bilayer, how molecules get in and out of the membrane. I can do that real quick before I go to sleep. One more thing. All right, so here's phospholipid bilayer. It's made out of phospholipids. Phospholipids have a polar head and non-polar tails. And awesome. Glad I could help. Good luck on the exam. Hope you don't feel nearly as cooked as you did when you started. So because there's a polar outside, they want to go towards the edge where there's water, and the non-polar insides want to stay toward one another. And so things that are non-polar usually don't have any problem getting across the membrane. Things that are polar will need some assistance to get across the membrane, and that assistance is usually in the form of like a protein, like a membrane protein, like transport protein. So yeah, you're still cooked. I mean, maybe not. Go watch those YouTube shorts, man, while you're going to sleep. Just thumb through them. There's some, just my voice is, it just puts you to sleep, and you'll just learn them that way. And it's good. That's why I made those, so people could just watch them really quick. Man, this has been great. Thank you guys for being such a great audience. I guess is not the right word. Class, maybe is better. Very civil, great questions. Hopefully you feel like you've been prepared. Please let me know in the comments of one of my videos how you did. how you feel like you did. After the exam, they're going to release the FRQs in a couple days, and then I will be creating videos to answer all the FRQs. So please check those out. But good luck on your test tomorrow.